Okay, let's go through this question. 6.1, give three structural differences between mRNA and a tRNA molecule. It's worth 3 marks. So why don't you pause the video, grab a pen, grab some paper and scribble down what are the differences. Okay, I'm going to go through the answers now and you can see how many you got right. There's about 5 or 6 different differences, but we'll go through them. So if you imagine DNA over here in pink, mRNA is a single short strand of a section of DNA. It's basically a copy of a gene, right? And what is tRNA? Well, tRNA is the carrier molecule that is going to deliver an amino acid in transcription to the ribosome. Now, tRNA has a different shape. How would you compare that blue drawing of mRNA to the shape of tRNA? How would you compare that? And what is attached to a tRNA molecule that isn't attached to an mRNA? So let's get some of these differences down. So first of all, dealing with the shape, if we look at the blue mRNA on the left, it's linear, meaning it's just basically a line, a straight piece. They're not joined at the ends. But if we look at the tRNA, it's clover shaped. It's basically a loop of DNA, isn't it? A circular piece of DNA joint at the ends, the green one there. Also, mRNA has codons, which is the nucleotide sequence, which is kind of opposite, but joined by complementary base pairing to the anticodons on the tRNA. So I'd be like mRNA has codons, tRNA has anticodons. And now the tRNA has an amino acid binding site, whereas the mRNA has no amino acids attached. Okay, let's go over a couple of other ones. So if we look internally within the tRNA, these are all nucleotides and there's intermolecular forces between them. That's a really rubbish drawing. Let me just rub that out. Excuse them. What? Okay, let's redo that. Okay, so here is our clover bit of tRNA. Now, what I'm trying to show you is that it will have like guanine on the inside and cytosine opposite, and they'll hydrogen bond to each other, hence why it's in a loop or a clover-shaped loop. Whereas the mRNA is linear because there is no complementary base pairing with another nucleotide. So there's no hydrogen bonds. Okay, looking at this next question, 6.2, table 3 shows mRNA codons and the amino acid coded by each codon. Okay, figure 5 shows the mRNA base sequence produced when part of a gene coding for an enzyme is transcribed. So what we're going to do is we're going to go through, it says use table 3 to give the amino acids coded by the mRNA base sequence in figure 5. So what we're going to go through here is try and work out which amino acid is coded for by each of these 3 triplet codons on the mRNA. So we've got UUU, CGG, and GCG. So we're really learning here how to read this kind of table 3, which is a very common table to be able to read when you're dealing with DNA. Okay, so let's have a look. We've got UUU, CGG, and GCG, and we need to work out what these are and stick them in these blank boxes. Let's do it now, folks. Right, so looking at the first one, you're going to circle the first U. So it means that the amino acid is found in this row. And then it's also, we look at the second base, which is also U. So it has to be in this column. So it's got to be PHE or LEU. Now, we can see the U is there, and we're going to go across, draw a line across. It has to be PHE because that third base is in line with that. So our first amino acid is PHE. We're now going to look at the second one is CGG, and that means because it's in this row where I've circled in green, it's one of these 5 amino acids. It has to be in that column, and it doesn't matter which one it was in that column. They would have all been arginine, ARG. Okay, now let's look at the third one. I'm going to pick another color. So we've got G first of all in brown here, then C, and that tells me it's in this row. It has to be in that column. It's going to have to be alanine. We don't even need to look at the last one. So it could have been G, could have been U, could have been whatever it was. The code GCG codes for alanine, but GCA or GCC would code for alanine as well. And there you go. It's a lot of work for 1 mark. Stingy AQA. 6.3. A mutation occurred in part of the gene transcribed in question 6.2. Figure 6 shows the mRNA base sequence produced when the identical part in the mutated gene is transcribed. So we've got UUU, AGG, GCG. The amino acid coded by this mRNA base sequence forms part of the enzyme's active site. Use all the information in the question to name the type of mutation that occurred to produce the mutated gene, give the change in the DNA caused by the mutation, explain the effect this mutation will have on the function of the enzyme. It's worth 4 marks, and they've spaced it out nicely for us like this. So what we need to do is zing back up and see what are the differences. And if we have a little look here, we've got UUU and UUU, so there's been no change, no mutation there. And we've got GCG and GCG, so no mutation here. However, in the third one, we got CGG, and this has turned to AGG. So there has been a swapping of one of the bases. C has swapped for A. So that is what has happened, and we call this a substitution mutation. You can call it a silent mutation as well, as we'll come on to with our explanation. Now, what is the change in the DNA? This is the bit I'm going to forget. Was it C got changed to A? I think it was. So on the second codon, the middle codon, a C was swapped to become an A. So the C base was swapped for an A. Let me just double bubble check. Yes, that's right. Now, what is our explanation of the effect on the function of the enzyme? So we're going to have a little look up here, and what we can see with CGG, that is arginine, right? Now we have gone to A. Oh God, I've done that wrong. Not ACG, you idiot. We're talking AGG. Whoopsie. Hold on. Let me delete this. Okay, what I'm trying to do is show you the comparison so we can actually work out what's happened. Has this changed the amino acid, or is it for the same amino acid? So you'll see here, instead of being CGG, it went to AGG. Let's do that again. A's up there, G's there. I'm doing it in pink. It also codes for arginine, right? So it doesn't matter the fact that this C swapped for an A. They both code for arginine. There we go. Finally got to the bottom of it. Sorry about that, folks. A little bit of a hiccup there. So it is a silent mutation. And what this means is, as DNA is degenerate, meaning that more than one codon there's more than one codon coding for an amino acid. Even though the C has been substituted for an A, they both still code for the amino acid arginine, both of these triplets. What does this mean in relation to the enzyme? Well, this means that the enzyme will have the exact same primary structure, excuse my spelling. What is the primary structure? That is the structure that is going to inform the type of bonding that we're going to have, because it gives you the type of R groups, and that will decide what type of ionic, covalent, hydrophilic, hydrophobic interactions or bonds you form in the tertiary structure. So it means that even though it's been mutated, it has the same tertiary structure, the same 3D shape. The enzyme's active site will still be exactly the same, and it will still be functional, and it will still form an enzyme substrate complex. So the enzyme will be fully working even though it has had a mutation. This is called a substitution. And now for the second mark here, it says the change in the DNA. Well, the change in the mRNA was C to A, so cytosine was swapped to adenine, and that means that in the complementary DNA template that coded for the mRNA, the swap was guanine to thymine. So they actually need you to say that G was swapped for T in the DNA, even though we're given here the mRNA C for A. Well, C would be complementary to guanine on the DNA. So normally guanine would produce the cytosine in the mRNA, but the guanine was changed to thymine. So when the mRNA was coded for, it was adenine instead of cytosine. Complex? Well, not that bad. All right, let's move on to the next question.
You're still here? It's over.